Hello, my name is Andrei Alyokhin, and I am a professor of applied entomology at the University of Maine. We work with different insects that affect humans. A lot of our work is with the Maine potato industry, and one of the big projects that we're currently involved in right now is studying potato virus Y and its aphid vectors. So the question is, why would entomologists study a virus? But the key to its spread between potatoes is vectoring by aphids. So there is a number of aphid species. Aphids are insects, as you hopefully know. So they feed on infected plants, then they move on to uninfected plants, and they bring virus with them. So while feeding by aphid itself is usually not a very big deal, at least in potatoes, virus transmission could be a big deal. So no aphids means no viruses, and if we can figure out what to do with aphids, then we will hopefully take care of viruses as well. So here is a plant that grew from a tuber that was infected with potato virus Y. You can see that it is significantly smaller than a neighboring plant which doesn't have a virus. Also, tubers that will be harvested from this plant may have some quality problems, such as necrotic rings, basically circles of dead dark tissue in them. They will also be smaller in size, and the more of those infected plants you have, the worse your harvest will be. Here is another plant that had virus. Also, you can see it's stunted and leaves are kind of mosaicy. And here is a white stake. That means that we deliberately planted an infected tuber here to study transmission and effects of different treatments on transmission. And these plants, they acquired virus during this season. You can see also that leaves are kind of funky. They're a little bit smaller, but they don't look as bad as that plant. However, there will still be problems with tubers and tuber yield. So during this season, most likely aphids carried virus from this plant that we introduced in the field to these plants that grew from tubers that originally did not have virus, but they acquired virus during this summer. This is a field with one of our experiments studying virus transmission by aphids. You can see there are small plots of potatoes which have different treatments applied to them, and there are strips of oats in between those plots, and this is to reduce transmission of virus between the plots, because different plots have different treatments, so we don't want aphids to jump from plot to plot to carry virus and mess up our experimental design. So if an aphid wants to leave this potato plot, chances are it will land somewhere around here and either stay here, or if it doesn't stay, it will still probe around grass, oats. Oats is a grass, and then lose virus in the process. So if this aphid moves here, it will not carry virus anymore, and it will not mess up our experimental treatments. Each plot has a yellow sticky card attached to it, so we can see what kind of insects are flying around, in particular what kind of aphids are flying around. Insects see yellow as we see green, so for them this is like a foliage. So when an insect, including aphids, wants to land, it will go down here and then it will get stuck, and we can bring it to the lab and identify it. You can see that this is mounted kind of flush with the level of leaves, so most insects would land on nearby plants, but some would go for the trap and stay here. This way we know what's coming into the fields when, and we also know, for example, if we don't see any virus transmission, which is unlikely, whether that happened because there was no aphids to begin with, or maybe our treatments worked so well that there was no virus left. Here is a part of another study on looking potatoes and virus interaction. So we have about 34 different varieties of potatoes. Some of them are commercially available, others are on different stages of development. And last year they were all challenged with aphids that we knew were infected with viruses, so we released those aphids on potatoes, then harvested tubers, planted them this year to see whether they acquired the infection and whether they show any symptoms. So if you look, for example, at this variety here, you can see that here is plant is a little bit smaller, and this mosaic on leaves, so the plant is infected. Probably looking at its symptoms are quite pronounced. It grew from a tuber that was infected last year. This one looks significantly better, but there are still some symptoms that may be showing up, so this may have been transmitted by some aphids this year, or maybe it just got less of it, I don't know. But if we look at the variety next to it, do not really see any symptoms. There is no mottling, there is no crinkling of leaves, there is no stunting. They look big and flush and green. So maybe this is something that's resistant to the virus. Hopefully it is resistant. However, maybe it also acquired virus and just does not express any symptoms. This is asymptomatic carrier, and this is not a good thing, because if those two varieties are grown on fields next to each other, then virus can jump from here to here. So by screening varieties, by looking at virus infection, at looking at symptoms, we can characterize which one of them are resistant to disease and which one of them are susceptible. And this is one of the considerations where and when to grow them. These are a couple of cages where we keep aphids to be used in experiments. And cage keeps natural enemies away, such as lady beetles or green lacewings, so aphids feel safe and sound inside, and they can build up their populations pretty quickly. So then we harvest some aphids from here, and then either release them on experimental plots, or maybe bring them to the lab and do some laboratory experiments. So let me remove this cage for now, and you can hopefully see some aphids over here. Okay, so the plant is basically packed with them, and some of them have wings and are ready to fly away, and these are especially bad in carrying virus around. Then some of them are wingless, but they still can transmit disease between plants which grow next to each other. So while winged aphids are important for carrying it to new places and establishing new outbreaks, wingless aphids may be important for spreading it within the field and for enlarging those outbreaks. Hopefully now you have some idea of what we are doing at our research program relative to potato viruses and their transmission by aphids. If you are interested in this system and if you're interested in learning more about viruses, about potatoes, about their management, then I would suggest that you watch this video on YouTube. We put it together last year. It's mostly for growers and other agricultural professionals, but I think it may be useful for some of you, at least for people who are interested in agriculture, in pest management, or maybe in disease transmission by insects in general. So please check it out, and hopefully you like it.